Hello there, and welcome to the Sleepy Science Society. I'm so glad you found your way here tonight. Maybe you've had a long day, or perhaps you're the kind of person who likes drifting off to the gentle rhythm of scientific wonder rather than counting sheep. Either way, you're in the right place. Tonight, we are embarking on a fascinating journey through one of the most remarkable systems in your body, a microscopic universe that's been protecting you since before you took your first breath. And if these quiet explorations help you unwind or give you a peaceful way to end your day, I'd love it if you'd consider liking this video and subscribing to the channel. It helps other curious souls find their way here too, one peaceful night at a time. Tonight, we're going to explore the immune system, not just as a collection of cells and organs, but as a living, breathing defense network that's constantly working to keep you alive. Right now, as you settle in and let your muscles relax, your immune system is waging thousands of microscopic battles. It's patrolling your bloodstream, standing guard at your skin, filtering threats from your lymph nodes, and remembering every pathogen it's ever encountered. It's scanning each of your trillions of cells, checking for signs of damage or invasion. It's a symphony of coordination happening entirely without your conscious thought. A guardian that never sleeps even when you do. What makes the immune system so extraordinary is not just its power, but its intelligence. It can distinguish your own healthy cells from billions of potential invaders. It can remember a virus you encountered decades ago and mount a defense within hours if it ever returns. It can adapt to threats it's never seen before, learning and evolving throughout your lifetime. And it does all of this while maintaining a delicate balance. Powerful enough to destroy dangerous pathogens, but restrained enough not to attack your own body. Over the next couple of hours, we'll journey through this hidden landscape together. We'll meet the organs that serve as training grounds and headquarters. We'll encounter the diverse cellular army that patrols your body. We'll witness the ancient, rapid-fire responses of innate immunity and the precise, targeted strikes of adaptive immunity. We'll discover how your body remembers every battle it's ever fought, and how that memory protects you for years or even a lifetime. We'll explore what happens when this system falters, when it attacks itself or fails to recognize danger. And we'll see how your daily choices, from the sleep you get to the food you eat, shape the effectiveness of this remarkable defense network. The immune system is one of the most complex biological systems known to science. A masterpiece of evolution refined over hundreds of millions of years. Yet it's also deeply personal, as unique to you as your fingerprint, shaped by every infection you've overcome, every vaccine you've received, every exposure you've survived. Understanding how it works is not just an intellectual exercise. It's a way of understanding yourself at the most fundamental level, of appreciating the constant, invisible work that keeps you alive. So let your breathing slow. Let your thoughts drift. Let the day's tensions melt away. We're about to descend into a world too small to see but too important to ignore. A universe of protection that exists within you, working tirelessly through every moment of every day. Tonight, we honor that work by learning its secrets. Let's begin. The invisible guardian. What is the immune system? The immune system is not a single organ or a simple structure. It's a vast, interconnected network that spans your entire body, a collaboration between organs, tissues, cells, proteins, and chemical signals, all working toward a single purpose: keeping you alive. Think of it as a combination of military force, intelligence agency, and communications network all rolled into one, operating at a microscopic scale with extraordinary precision. At its most fundamental level, the immune system exists to distinguish self from non-self, to recognize what belongs in your body and what doesn't. This is a far more challenging task than it might initially seem. Your body contains trillions of cells, each carrying specific molecular signatures that identify them as yours. Meanwhile, you are constantly exposed to trillions of bacteria, viruses, fungi, parasites, and other potential threats. Some of these invaders are genuinely dangerous, capable of making you sick or even killing you. Others are harmless or even beneficial. The immune system must make split-second decisions about which to attack and which to tolerate, all while avoiding the catastrophic errors of attacking your own healthy tissue. This system accomplishes its mission through multiple overlapping layers of defense, each more sophisticated than the last. The outermost layer consists of physical and chemical barriers that prevent most threats from ever entering your body in the first place. Your skin forms a nearly impenetrable wall, secreting oils and acids that kill many microbes on contact. The mucous membranes lining your respiratory and digestive tracts trap particles and sweep them away. Tears, saliva, and stomach acid all contain antimicrobial chemicals that destroy potential invaders before they can establish a foothold. When something does breach these barriers, it encounters the innate immune system, your body's rapid response force. This system is ancient in evolutionary terms, present in some form in nearly all multicellular organisms. It responds within minutes to hours, deploying cells that can recognize broad patterns common to many pathogens. These cells don't need prior exposure to know something is wrong. They're programmed to detect molecular signatures that don't belong, danger signals that indicate infection or tissue damage. When they find these signals, they attack, immediately, engulfing invaders, releasing toxic chemicals, and triggering inflammation to call for backup. But the innate system, powerful as it is, has limitations. It can't adapt to new threats, can't learn from experience, and can't provide the kind of specific targeted response that some infections require. That's where the adaptive immune system comes in, a more recent evolutionary innovation found only in vertebrates. This system takes longer to activate, requiring days rather than hours. But it compensates with extraordinary precision and memory. It generates cells and antibodies tailored to specific pathogens, like custom-designed weapons for each particular threat. And once it defeats an invader, it remembers that enemy for years, sometimes for life, enabling it to respond almost instantly if that pathogen ever returns. These two branches, innate and adaptive, don't work in isolation. They're in constant communication, each one activating and amplifying the other. The innate system serves as the first responder and the alarm system, alerting the adaptive system to danger. The adaptive system, in turn, can enhance and direct the innate response, making it more effective and more focused. Together, they form a defense network of staggering complexity, one that medical science is still working to fully understand. The immune system's job extends beyond fighting infection. It also clears away dead and damaged cells, like a cleanup crew working constantly to maintain order. It monitors for cancer cells, abnormal cells that could grow into tumors if left unchecked, and destroys them before they can spread. It helps wounds heal by orchestrating the complex process of tissue repair. It even plays a role in pregnancy, helping the mother's body tolerate the genetically distinct fetus rather than rejecting it as foreign tissue. What makes this system so remarkable is its ability to learn and remember while maintaining tremendous diversity and flexibility. Your immune system can potentially recognize and respond to billions of different molecular patterns, far more than the number of genes in your entire genome. It achieves this through clever genetic tricks, randomly recombining pieces of DNA to generate vast libraries of receptors, each one unique. Most of these receptors will never encounter their matching pathogen, but having such diversity ensures that when a new threat emerges, you almost certainly have some cells capable of recognizing it. At the same time, the immune system must walk a constant tightrope between vigilance and restraint. Too little activity, and infections overwhelm you. Too much, and the system attacks your own tissues, causing autoimmune diseases. Or it overreacts to harmless substances, triggering allergies. The system must be powerful enough to stop deadly pathogens, but careful enough to distinguish between a dangerous bacterium and a beneficial gut microbe, between a cancer cell and a healthy one, between a foreign invader and your own tissue. This balance is maintained through multiple checkpoints and regulatory mechanisms. Certain immune cells specialize in dampening responses, preventing the system from spiraling out of control. Molecular signals provide feedback, ramping activity up or down depending on what's needed. The system constantly calibrates itself, adjusting its sensitivity based on the threats it encounters and the state of your body. And here's something truly fascinating. Your immune system is uniquely yours. While the basic architecture is the same in all humans, the specific collection of memory cells, the particular antibodies circulating in your blood, the exact balance of different cell types, all of these vary based on your individual history of exposures. Two people might have genetically identical immune systems at birth, but after a lifetime of different infections, vaccinations, and environmental exposures, their immune profiles would be as distinct as their faces. Your immune system is a living record of everywhere you've been and everything you've encountered. Scientists estimate that your immune system contains trillions of cells, spread throughout your body but concentrated in specific organs and tissues. These cells are constantly in motion, patrolling through blood vessels and lymphatic channels, surveying tissues, gathering in lymph nodes to exchange information. They communicate through an intricate language of chemical signals, hundreds of different molecules that carry specific messages. Danger here, need help, stand down, remember this. The coordination required to manage all of this activity, to mobilize the right cells to the right place at the right time, is staggering. And all of this happens automatically, beneath your awareness, every second of every day. You don't consciously direct your immune cells to attack that virus or remember that vaccine. You don't decide when to trigger inflammation or when to call it off. The system operates itself, a masterpiece of self-organization that required hundreds of millions of years to evolve, but that now runs seamlessly, without any input from your conscious mind. As we drift deeper into this topic, we'll explore each component of this system in detail. We'll meet the organs that serve as its command centers and training facilities. We'll encounter the diverse array of cells that carry out its missions. We'll witness the elegant choreography of an immune response, from first detection to final resolution. We'll see how memory forms and why vaccination works. And we'll discover what happens when this delicate system malfunctions. But for now, just appreciate the magnitude of what's happening inside you at this very moment. While you rest, your immune system stands watch, a tireless guardian in a battle that never ends. The architecture of defense, organs, and structures. To understand the immune system, we need to explore its physical infrastructure, the organs, and tissues where immune cells are born, trained, stored, and deployed. Unlike the heart or lungs, which are single, centralized organs, the immune system is distributed throughout your entire body. Its components form a network connected by blood vessels and a parallel circulatory system called the lymphatic system, allowing immune cells to travel anywhere they're needed. Let's start at the beginning, with the birthplace of nearly all immune cells. The bone marrow. Deep inside your bones, in the soft, spongy tissue at their core, something remarkable is happening. Every second of every day, your bone marrow produces millions of new blood cells through a process called hematopoiesis. These include the red blood cells that carry oxygen and the platelets that help your blood clot. But most relevant to our story are the white blood cells, the warriors and scouts of your immune system. All of these cells arise from a small population of hematopoietic stem cells, master cells with the potential to become any type of blood cell. Through a series of carefully controlled divisions and differentiations, these stem cells give rise to the full spectrum of immune cells. Some become myeloid cells, which include the rapid response troops of innate immunity. Others become lymphoid cells, which mature into the specialized forces of adaptive immunity. This continuous production is essential because many immune cells have relatively short lifespans, ranging from hours to months. Your body must constantly replenish its defenses. The bone marrow is not just a factory, it's also a sanctuary for certain long-lived immune cells called plasma cells, which secrete antibodies continuously, providing persistent protection against pathogens you've encountered before. These cells can survive in the bone marrow for years, sometimes decades, quietly producing the molecular weapons that keep old enemies at bay. From the bone marrow, newly formed immune cells enter the bloodstream and travel to other organs where they complete their education. One of the most important of these is the thymus, a small, butterfly-shaped organ located behind your breastbone. The thymus serves as a finishing school specifically for T cells, one of the most crucial cell types in adaptive immunity. T cells arrive at the thymus as immature precursors, and undergo a rigorous selection process. They're tested to ensure they can recognize foreign molecules but don't react strongly to your own body's proteins. This is critical because T cells are powerful and potentially dangerous. A T cell that attacks your own tissue could trigger devastating autoimmune disease. So, the thymus imposes strict quality control. Cells that fail the test, either because they can't recognize anything or because they react to self-proteins, are eliminated. Only about 2% of T cells survive this selection, but those that do are ready to serve as guardians, capable of distinguishing self from non-self with high reliability. The thymus is most active during childhood and adolescence, producing vast numbers of T cells as your immune repertoire develops. As you age, the thymus gradually shrinks, its tissue replaced by fat. This decline is one reason why elderly individuals often have weaker immune responses. However, the thymus never completely stops functioning. Even in older adults, it continues producing some new T cells, supplementing the population of long-lived memory T cells that have accumulated over a lifetime. Once immune cells complete their maturation, many take up residence in lymph nodes, small, bean-shaped structures scattered throughout your body. You have hundreds of lymph nodes clustered in your neck, armpits, groin, and deep within your abdomen and chest. They're connected by a network of lymphatic vessels that parallel your blood vessels, forming a separate circulatory system for immune surveillance. Lymph nodes serve as filtering stations and meeting places for immune cells. Fluid from your tissues, called lymph, drains into these vessels and flows through the nodes, where immune cells scrutinize it for signs of trouble. If you've ever had swollen lymph nodes during an infection, you've felt this system in action. The swelling indicates that immune cells are gathering in that node, multiplying and coordinating a response to a threat detected nearby. The nodes act as regional command centers, places where cells can present antigens they've captured, recruit help, and launch coordinated attacks. The architecture of a lymph node is intricate and purposeful. The outer regions, called follicles, are packed with B cells, the antibody-producing specialists. The inner regions contain T cells and antigen-presenting cells. When a threat is detected, these areas become hives of activity, with cells dividing rapidly and antibodies being produced in vast quantities. Once the threat is neutralized, the node returns to a quieter state, but some of the activated cells remain as memory cells, ready to respond if that particular threat ever returns. Another crucial organ is the spleen, located in your upper left abdomen. The spleen is the body's largest lymphoid organ, and it serves multiple functions. As blood filters through the spleen, it passes through specialized tissue containing dense populations of immune cells. These cells monitor the blood for pathogens and abnormal cells, much as lymph nodes monitor tissue fluid. The spleen also stores platelets and white blood cells, maintaining reserves that can be quickly deployed during infection or injury. In addition, the spleen has a unique role in managing red blood cells. It identifies and removes old or damaged red blood cells from circulation, recycling their components. This housekeeping function is important for maintaining healthy blood and preventing the buildup of cellular debris. People who lose their spleen due to injury or disease can survive, but they're at higher risk for certain infections, particularly those caused by encapsulated bacteria, demonstrating the spleen's importance in immune defense. Your tonsils and adenoids, located at the back of your throat and nasal passages respectively, are also part of the immune system. These structures are strategically positioned at entry points where many pathogens try to invade, through the mouth and nose. They contain lymphoid tissue packed with immune cells that serve as an early warning system. When you have a throat infection and your tonsils become swollen and painful, that's evidence of immune cells fighting back against invading bacteria or viruses. But not all immune tissue is organized into discrete organs. Much of your immune system is diffusely distributed throughout your body, particularly in mucosal surfaces. These are the moist linings of your respiratory tract, digestive system, and genitourinary tract. Collectively, mucosal surfaces represent one of your body's largest interfaces with the external environment, and they're heavily defended. Mucosa-associated lymphoid tissue contains scattered collections of immune cells, always ready to respond to threats. Your gut alone contains more immune cells than any other part of your body, a reflection of the enormous microbial populations that live there. The gut immune system must perform a delicate balancing act, tolerating beneficial bacteria that help with digestion while attacking pathogens that cause disease. This requires sophisticated regulatory mechanisms that are still not fully understood. Your skin, though not typically thought of as an immune organ, also plays a crucial defensive role. Beyond serving as a physical barrier, skin contains specialized immune cells called Langerhans cells, which can capture antigens and present them to other immune cells, initiating adaptive immune responses. Skin also produces antimicrobial peptides, small proteins that kill bacteria and fungi on contact. When you get a cut or scrape, these cells and molecules work quickly to prevent infection. All of these structures, from bone marrow to lymph nodes to mucosal tissue, are interconnected by the lymphatic system. Lymphatic vessels form a one-way network that drains fluid from your tissues, filters it through lymph nodes, and eventually returns it to your bloodstream. This system serves multiple purposes. It maintains fluid balance, transports fats from the digestive system, and, most importantly for our purposes, provides a highway for immune cells and antigens. Immune cells constantly circulate between blood and lymph, patrolling tissues and gathering in lymphoid organs. A single lymphocyte might make a complete circuit through your body every few hours, checking in at lymph nodes, surveying tissues, returning to blood. This constant traffic ensures that immune cells can be where they're needed quickly, and that information about threats can be shared throughout the system. The lymphatic system also provides a route by which infections can spread, which is why swollen lymph nodes can sometimes appear in chains as infection progresses from one node to the next. But more often, the nodes successfully contain and eliminate threats, preventing them from reaching the bloodstream where they could cause systemic infection. What's remarkable about this anatomical architecture is how it balances centralization with distribution. Certain functions, like the production of new immune cells and the training of T cells, are centralized in specific organs. But the deployment of immune defense is highly distributed, with immune cells present throughout every tissue in your body. This combination ensures both efficiency and comprehensive coverage. No matter where a pathogen tries to invade, immune cells are ready to respond. This infrastructure is also dynamic, not static. Lymphoid tissue can expand and contract based on immune activity. During an infection, lymph nodes can swell significantly as cells multiply. New lymphoid follicles can form in tissues experiencing chronic inflammation. The system adapts its physical structure to meet demand, like a city expanding its infrastructure during population growth. As you lie there relaxing, this vast network is humming with activity. Lymph is slowly draining from your tissues, carrying cellular debris and potential threats to nearby lymph nodes. Immune cells are percolating through your bone marrow, maturing and entering circulation. Your spleen is filtering your blood, removing old cells and checking for abnormalities. Cells in your gut are monitoring the trillions of bacteria that live there, maintaining the peace. And all of this happens automatically, orchestrated by chemical signals and cellular interactions that require no conscious oversight. Understanding this architecture helps us appreciate the immune system not as an abstract concept, but as a physical reality. A tangible network of organs and cells distributed throughout your body, all working in concert to keep you healthy. In the sections ahead, we'll meet the cellular inhabitants of these organs, the diverse populations of immune cells that carry out the system's missions. We'll see how they're trained, what they do, and how they coordinate their activities. But first, we need to appreciate the stage on which this drama unfolds, the organs and tissues that make immune defense possible. The cellular army, white blood cells, and their roles if the organs we just discussed are the infrastructure of immune defense, then white blood cells, or leukocytes, are the system's active agents, the soldiers, scouts, and strategists that carry out its missions. These cells are astonishingly diverse, each type specialized for particular tasks, working together in complex combinations to detect, attack, and remember pathogens. White blood cells are present throughout your body but can concentrate rapidly where they are needed. A single drop of blood contains thousands of them, and the total number in your body at any given time numbers in the billions or even trillions. What's remarkable is not just their abundance, but their variety. Medical science recognizes dozens of different types of white blood cells, each with distinct features, functions, and lifespans. Let's start with neutrophils, the most abundant type of white blood cell in your bloodstream. Neutrophils are the infantry of your immune system, fast-moving cells that rush to sites of infection or injury. When you cut yourself and bacteria enter the wound, neutrophils arrive within minutes, drawn by chemical signals released by damaged tissue. These cells are professional killers, equipped with an arsenal of antimicrobial weapons. Neutrophils employ several strategies to eliminate threats. They can engulf bacteria in a process called phagocytosis, literally eating the invaders and destroying them with toxic chemicals inside specialized compartments. They can also release nets of DNA studded with antimicrobial proteins, trapping bacteria like flies in a spider's web. And in some situations, they release their toxic contents directly into the surrounding space, creating a hostile environment for microbes, though this can also damage your own tissue if not carefully controlled. The life of a neutrophil is brief and intense. These cells survive only a few hours to a few days, spending most of that time patrolling the bloodstream or tissues. When they encounter infection, they fight until they're exhausted, then die, often forming the pus that accumulates in infected wounds. Despite their short lives, neutrophils are produced in vast numbers. Your bone marrow generates about 100 billion new neutrophils every day, maintaining a constant supply of fresh troops. Macrophages are another critical cell type, larger and longer-lived than neutrophils but with similar capabilities. The name literally means big eater, and that's exactly what they do. Macrophages engulf and digest not just bacteria, but also dead cells, cellular debris, and other waste products. They're the cleanup crew and the sentinels, constantly surveying their environment for anything that doesn't belong. But macrophages do more than just eat. They also serve as antigen-presenting cells, displaying fragments of the pathogens they've consumed on their surface. This presentation alerts other immune cells, particularly T cells, to the presence of infection and helps coordinate adaptive immune responses. Macrophages also secrete chemical signals called cytokines that regulate inflammation and recruit other immune cells to the area. In this way, they act as coordinators and communicators, orchestrating the immune response. Macrophages are found throughout your body, but they're especially concentrated in tissues that interface with the external environment, like the lungs, gut, and skin. Different tissues have specialized populations of macrophages with names like Kupffer cells in the liver and microglia in the brain. These cells are adapted to their specific locations, performing local cleanup and surveillance while maintaining the capacity to fight infection when necessary. Dendritic cells, despite their name, are not neurons but immune cells with long branching projections that resemble the dendrites of nerve cells. These projections increase their surface area, helping them capture antigens efficiently. Dendritic cells are arguably the most important antigen-presenting cells in your body, serving as the bridge between innate and adaptive immunity. These cells reside in tissues throughout your body, especially at potential entry points for pathogens. When they encounter something suspicious, they capture it and travel to the nearest lymph node, where they present what they've found to T cells. This antigen presentation is how adaptive immune responses are initiated. T cells can't recognize pathogens on their own. They need dendritic cells to show them what to look for. Once activated by a dendritic cell, T cells multiply and differentiate, launching a targeted immune response. Dendritic cells also help maintain tolerance to harmless substances and beneficial microbes. In the absence of danger signals, they present antigens in a way that promotes tolerance rather than activation. This dual function, promoting immunity to threats and tolerance to harmless substances, makes dendritic cells crucial regulators of immune balance. Natural killer cells, often abbreviated NK cells, are part of the innate immune system but have some features reminiscent of adaptive immunity. They're called natural killers because they don't need prior activation or training to attack target cells. Instead, they constantly survey other cells for signs of trouble, looking for infected cells or cells that have become cancerous. NK cells recognize their targets through an elegant system of activating and inhibiting signals. Healthy cells display surface molecules that tell NK cells to stand down. But when cells become infected or turn cancerous, they often lose these inhibitory signals or display stress-induced activating signals. NK cells detect these changes and attack, releasing toxic granules that cause the target cell to undergo programmed death. This system provides rapid defense against viruses and cancer without requiring the days or weeks needed to mount an adaptive response. Interestingly, recent research has shown that NK cells, like adaptive immune cells, can form a kind of memory after certain infections, responding more vigorously upon re-exposure. This discovery has challenged traditional ideas about the strict boundary between innate and adaptive immunity, suggesting that the immune system is even more flexible and sophisticated than previously thought. Mast cells and basophils are closely related cells that play important roles in allergic reactions and defense against parasites. They're packed with granules containing histamine and other inflammatory mediators. When activated, they dump these contents into the surrounding tissue, causing the symptoms we associate with allergies: swelling, redness, itching, mucus production. While these reactions can be unpleasant and even dangerous when triggered inappropriately, they evolved as defense mechanisms against parasitic worms, which are too large for immune cells to engulf. The inflammatory response triggered by mast cells makes the local environment inhospitable for parasites and can physically expel them through coughing, sneezing, or diarrhea. However, in modern developed countries where parasitic infections are rare, mast cells often respond to harmless substances like pollen or pet dander, causing allergies that reduce quality of life without providing any benefit. Eosinophils are another cell type specialized for fighting parasites. They're named for their appearance under a microscope, where they pick up a characteristic pink color from a dye called eosin. Eosinophils can attach to parasites and dump toxic proteins onto their surface, damaging the parasite's protective covering. Like mast cells, eosinophils are also involved in allergic reactions, particularly asthma, where their activity contributes to airway inflammation. All the cells we've discussed so far are part of the innate immune system. But now we come to the lymphocytes, the specialized cells of adaptive immunity. Lymphocytes are smaller than most other white blood cells but more numerous, making up about 20 to 40% of white blood cells in the blood. There are two main types: B cells and T cells, each with distinct but complementary functions. B cells are the antibody factories of your immune system. When a B cell encounters its specific antigen, it becomes activated and transforms into a plasma cell, a specialized factory that churns out thousands of antibody molecules per second. These antibodies are Y-shaped proteins that bind to specific molecular patterns on pathogens, marking them for destruction, neutralizing them directly, or activating other immune components. Each B cell produces antibodies of one specificity, recognizing one particular molecular structure. Your body contains millions of different B cells, collectively capable of recognizing billions of different antigens through the clever genetic recombination we mentioned earlier. When infection occurs, The few B cells that recognize that particular pathogen multiply rapidly, generating an army of identical cells all producing the same antibody. This clonal expansion is how the adaptive immune system generates such massive, specific responses to individual threats. T cells are more diverse than B cells, with several functionally distinct subsets. Helper T cells, marked by a surface protein called CD4, coordinate immune responses by releasing cytokines that activate and direct other immune cells. Different subsets of helper T cells promote different types of responses, depending on the nature of the threat. Some subsets enhance antibody production by B cells. Others activate macrophages to destroy intracellular bacteria more effectively. Still others promote inflammation to combat certain parasites. Cytotoxic T cells, also called killer T cells, are marked by a surface protein called CD8. These cells directly attack infected cells or cancer cells, recognizing them through antigens displayed on their surface. When a cytotoxic T cell confirms that a target cell is infected or abnormal, it delivers a lethal cocktail of enzymes that trigger the cell's self-destruct program. This eliminates the infected cell before viruses inside can replicate and spread, helping contain infections. Regulatory T cells, another subset, have the opposite function. They suppress immune responses rather than promoting them. This might seem counterproductive, but it's essential for preventing excessive inflammation and autoimmune reactions. Regulatory T cells help shut down immune responses once a threat is eliminated, preventing collateral damage to healthy tissue. They also help maintain tolerance to harmless antigens and beneficial microbes. People with defective regulatory T cells often develop severe autoimmune diseases, demonstrating how critical immune restraint is for health. All these cell types, from neutrophils to lymphocytes, must work together in coordinated ways to mount effective immune responses. They don't operate independently, but constantly communicate through direct cell-to-cell contact and chemical signals. A single immune response might involve dozens of cell types, each playing a specific role at a specific time, all orchestrated through feedback loops and regulatory mechanisms that science is still working to fully understand. What's particularly fascinating is how these cells make decisions. Despite lacking brains or consciousness, immune cells exhibit behaviors that look remarkably intelligent. They can sense chemical gradients and follow them to infection sites. They can distinguish friend from foe. They can remember past encounters. They can coordinate complex, multi-step attack sequences. All of this emerges from molecular interactions, from proteins binding to receptors, genes being turned on and off, signaling cascades activating and deactivating. The appearance of purposeful behavior arises from chemistry, from the physical properties of molecules following the laws of biochemistry. As you rest tonight, trillions of these cells are circulating through your body, each one a microscopic machine carrying out its programmed functions. Neutrophils are squeezing through blood vessel walls, following chemical trails to sites of minor tissue damage you are not even aware of. Macrophages are prowling through your tissues, engulfing debris and checking for intruders. Dendritic cells are sampling the environment, ready to sound the alarm if needed. NK cells are examining other cells, checking for signs of infection or malignancy. And lymphocytes are drifting through your bloodstream and lymphatic system, waiting for the antigen they're destined to recognize, a molecular signal that might come today or never. This cellular army, diverse and dynamic, forms the operational core of your immune defense. In the sections ahead, we'll see how these cells are deployed in actual immune responses, how innate and adaptive immunity work together, and how the system learns and remembers. But for now, simply appreciate the complexity and diversity of these cellular forces, each type uniquely equipped for its role, all working in concert to protect you from harm. The first line: innate immunity. Innate immunity is your body's rapid response system, the first line of defense that springs into action within minutes of detecting a threat. Unlike adaptive immunity, which we'll explore later, innate immunity doesn't require prior exposure to a pathogen or time to mount a response. It's ready from birth, programmed by evolution to recognize common features shared by many pathogens, and to respond with swift, if somewhat nonspecific, countermeasures. The first and most important component of innate immunity is physical barriers. Your skin is a formidable wall that keeps most microbes out simply by existing. Composed of layers of tightly packed cells, skin forms a waterproof barrier that few pathogens can penetrate when intact. The outer layer consists of dead cells filled with a tough protein called keratin, which makes the surface even more resistant to invasion. Skin also secretes oils and sweat that create a slightly acidic environment, inhibiting the growth of many bacteria and fungi. But your body has many openings that can't be covered by skin, places where the inside meets the outside. These are lined with mucosal membranes, moist tissues that serve as both barriers and active defense systems. The mucosa of your respiratory tract, for instance, produces sticky mucus that traps inhaled particles and microbes. Tiny, hair-like structures called cilia beat rhythmically, sweeping the mucus upward toward your throat where it can be coughed out or swallowed. This mucociliary escalator clears your airways of countless potential pathogens every day, keeping them from reaching your delicate lung tissue. Your digestive tract employs different defensive strategies. The highly acidic environment of your stomach kills most microbes that survive the antimicrobial enzymes in your saliva. Beneficial bacteria that colonize your intestines compete with pathogenic invaders for space and resources, a form of defense by occupation. The intestinal lining also produces antimicrobial peptides, small proteins that punch holes in bacterial membranes. And if a pathogen manages to adhere to the intestinal wall, rapid turnover of the intestinal lining cells can slough it off before infection establishes. Even your tears and saliva contain defensive molecules. Lysozyme, an enzyme found in many body fluids, breaks down bacterial cell walls. Lactoferrin binds iron, depriving bacteria of an essential nutrient. Defensins, which we mentioned before, directly kill microbes through their antimicrobial properties. These chemical defenses work continuously, maintaining a hostile environment for potential invaders at all the body's interfaces with the outside world. When a pathogen does breach these barriers, it triggers innate immune responses that begin within minutes. The key to innate immunity's speed is its use of pattern recognition receptors, proteins that detect molecular patterns common to many pathogens but absent from human cells. These patterns include components of bacterial cell walls, viral genetic material, and fungal cell walls. Because these molecular signatures are shared by large groups of microbes and are essential to their survival, pathogens can't easily evolve to hide them. This makes pattern recognition receptors reliable sensors for detecting infection. When pattern recognition receptors detect their targets, they activate the cells that carry them, triggering immediate defensive responses. Phagocytes like neutrophils and macrophages start engulfing microbes. Cells release inflammatory mediators that alter blood flow and make blood vessels leakier, allowing immune cells and defensive proteins to enter infected tissue more easily. Cells also release cytokines, chemical messengers that alert other immune cells to the danger and recruit reinforcements. This brings us to inflammation, perhaps the most familiar manifestation of innate immunity. When you get a cut or infection, the affected area becomes red, swollen, warm, and painful. These 4 cardinal signs of inflammation recognized since ancient times reflect the body's defensive response. Redness and warmth result from increased blood flow to the area, bringing more immune cells and oxygen to fight infection. Swelling occurs because blood vessels become more permeable, allowing fluid and proteins to leak into tissue. Pain results from chemical mediators that sensitize nerve endings and from pressure from the swelling itself. Inflammation, though unpleasant, is typically beneficial. The increased blood flow and vascular permeability help deliver immune cells to the site of infection rapidly. The fluid that accumulates dilutes toxins and brings in antibodies and complement proteins. The pain encourages you to rest the affected area, promoting healing. However, inflammation can also cause collateral damage. The toxic chemicals that immune cells use to kill pathogens can harm your own tissue if released carelessly. And if inflammation becomes chronic, persisting for months or years, it can contribute to serious diseases like arthritis, cardiovascular disease, and even cancer. The complement system is another crucial component of innate immunity, a cascade of proteins that can destroy pathogens and amplify other immune responses. Complement proteins circulate in the blood in inactive forms. When activated by the presence of pathogens or by antibodies bound to pathogens, they trigger a chain reaction where each protein activates the next, rapidly amplifying the signal. This cascade culminates in the formation of the membrane attack complex, a structure that punches holes in bacterial membranes, causing the bacteria to burst. But complement also generates protein fragments that recruit immune cells, coat pathogens to make them easier for phagocytes to engulf, and directly trigger inflammation. The complement system is thus both a weapon and a signaling system, attacking pathogens directly while also alerting and activating other immune components. Fever is another innate immune response, one that affects your whole body rather than just the site of infection. When certain cytokines released during infection reach the brain, they cause the hypothalamus to raise your body's temperature set point. This triggers responses that generate and conserve heat, shivering, vasoconstriction, seeking warmth. The resulting fever, while uncomfortable, has several benefits. Many pathogens grow more slowly at elevated temperatures, giving your immune system more time to contain them. Fever also speeds up immune cell activity and enhances some immune responses. However, very high fevers can be dangerous, and the body has mechanisms to prevent temperature from rising too high. Innate immunity also includes surveillance for cells that have become abnormal. Natural killer cells, which we discussed earlier, constantly check other cells for signs of infection or malignancy. This surveillance is crucial for catching problems early, before they can spread or worsen. Cells that die normally through programmed cell death or apoptosis do so quietly, in ways that don't trigger inflammation. But cells that die from injury or infection release danger signals that activate innate immunity, alerting the system that something is wrong. One of innate immunity's most important functions is activating adaptive immunity. Dendritic cells and macrophages that encounter pathogens in tissues capture antigens and carry them to lymph nodes, where they present them to T and B cells. The presence of danger signals, detected by pattern recognition receptors, determines whether this presentation activates adaptive immunity or promotes tolerance. In this way, innate immunity serves as the gatekeeper, deciding which threats warrant a full adaptive response and which can be handled by innate defenses alone. Recent research has revealed that innate immunity is more sophisticated than once thought. For instance, some innate immune cells can undergo epigenetic changes after encountering pathogens, making them respond more vigorously to subsequent infections. This phenomenon, called trained immunity, provides a form of memory in the innate system, though it's less specific and shorter-lived than adaptive immunological memory. This discovery has blurred the traditional distinction between innate and adaptive immunity, suggesting they form more of a continuum than a sharp divide. The innate immune system is ancient, present in some form in all multicellular organisms. Even plants and insects have innate immunity, though they lack adaptive immune systems. This antiquity speaks to innate immunity's fundamental importance. For most organisms, and for most of evolutionary history, innate immunity was the only immune defense available. It's remarkably effective against most common threats, clearing the majority of infections before they become serious. In humans, innate immunity remains the first responder and often the only responder needed. Most minor infections, most wounds, most exposures to pathogens are handled entirely by innate defenses without ever activating adaptive immunity. It's only when innate immunity is overwhelmed or when it encounters particularly sophisticated pathogens that adaptive immunity must be called in. As you drift towards sleep, your innate immune system is maintaining constant vigilance. At every interface between your body and the environment, physical and chemical barriers are holding the line. Pattern recognition receptors are scanning for danger signals. Phagocytes are patrolling tissues, ready to respond to any breach. And if infection does begin, the complex choreography of inflammation stands ready to contain it, to bring reinforcements, and if necessary, to activate the powerful, precise, but slower adaptive immune response we'll explore next, the specialists, adaptive immunity. While innate immunity provides rapid, general responses to common threats, adaptive immunity offers something far more remarkable: the ability to mount highly specific attacks against particular pathogens, and to remember those enemies for years, sometimes for life. This system, found only in vertebrates, represents one of evolution's most sophisticated solutions to the challenge of surviving in a world filled with microscopic threats. The defining feature of adaptive immunity is its specificity. Unlike innate immune cells that recognize broad patterns common to many pathogens, adaptive immune cells, specifically B and T lymphocytes, recognize individual molecular structures with exquisite precision. Each lymphocyte carries receptors specific for one particular antigen, one particular molecular shape. Your body contains millions of different lymphocytes, collectively capable of recognizing virtually any foreign molecule you might encounter. This tremendous diversity arises through a remarkable genetic mechanism. The genes encoding lymphocyte receptors don't exist in their final form in your DNA. Instead, they exist as libraries of gene segments that are randomly shuffled and recombined during lymphocyte development. Each developing lymphocyte cuts and splices its receptor genes differently, generating a unique receptor. With multiple gene segments to choose from and several mechanisms for introducing additional variation, this process can generate billions of different receptors from just a few hundred gene segments. This random generation creates a problem. Some randomly generated receptors will recognize your own body's proteins rather than foreign antigens. If such self-reactive lymphocytes were allowed to function, they would attack your own tissues, causing autoimmune disease. To prevent this, developing lymphocytes undergo selection in the thymus and bone marrow, testing their receptors against self-antigens. Cells that react too strongly to self are eliminated. Cells that can't react to anything are also eliminated. Only cells with receptors that strike the right balance survive, forming the repertoire of mature lymphocytes that patrol your body. Even after this selection, each type of lymphocyte specific for a particular antigen is present in very small numbers, perhaps only a few hundred or thousand cells out of billions. This creates another problem. How can so few cells generate a response powerful enough to fight infection? The answer is clonal expansion. When a lymphocyte encounters its specific antigen, it begins dividing rapidly, creating thousands or millions of identical daughter cells, all bearing the same receptor, all targeting the same enemy. This expansion takes several days, which is why adaptive immune responses are slower than innate responses. But the result is a massive, laser-focused attack on the invading pathogen. Let's examine how this works for B cells, the antibody-producing specialists. B cells recognize antigens directly through receptors on their surface, receptors that are actually membrane-bound versions of the antibodies they'll eventually secrete. When a B cell's receptor binds to its specific antigen, the cell becomes activated, but usually not fully. For most B cells, full activation requires help from T cells, a safeguard that prevents inappropriate responses. Once fully activated, a B cell begins the process of clonal expansion, dividing repeatedly to produce a large population of daughter cells. Some of these become plasma cells, specialized antibody factories. A single plasma cell can secrete thousands of antibody molecules per second, and with millions of plasma cells active during an infection, antibody concentrations in the blood can increase 1,000-fold or more. Antibodies are Y-shaped proteins with two functional regions. The tips of the Y's arms are the antigen binding sites, each one specific for the antigen that triggered the B cell. These binding sites recognize and attach to pathogens or toxins. The stem of the Y is the constant region, which interacts with other immune components to execute various defensive functions. Antibodies defend you in several ways. They can neutralize pathogens, binding to them in ways that prevent them from attaching to or entering your cells. They can opsonize pathogens, coating them with a molecular tag that makes phagocytes much more efficient at engulfing them. They can activate complement, triggering the cascade that punches holes in bacterial membranes, and they can cause pathogens to clump together, making them easier to filter out and destroy. The specific defensive mechanism depends partly on which class of antibody is produced, something B cells can switch between depending on the nature of the threat. T cells operate differently from B cells. While B cells recognize intact antigens directly, T cells can only recognize antigens that have been processed and presented by other cells. Antigen presentation occurs when a cell displays fragments of proteins on its surface, bound to proteins called MHC molecules. Nearly all your cells have MHC molecules on their surface displaying samples of the proteins inside them. This allows T cells to monitor what's happening inside cells, detecting infections that might otherwise be hidden from immune surveillance. Helper T cells recognize antigens presented by specialized antigen-presenting cells like dendritic cells and macrophages. When a helper T cell encounters its specific antigen properly presented, and when certain co-stimulatory signals are also present, the T cell becomes activated. It then begins dividing and releasing cytokines, chemical signals that orchestrate other immune responses. Different subsets of helper T cells release different cytokines, promoting different types of responses depending on the nature of the threat. Some helper T cells promote antibody production by B cells, providing the help that most B cells need for full activation. Others activate macrophages, making them better at killing intracellular pathogens. Still others promote inflammation or recruit specific types of immune cells to infection sites. Through these various functions, helper T cells act as coordinators, directing the immune response and ensuring that the right tools are deployed against each particular threat. Cytotoxic T cells function as assassins, seeking out and destroying infected cells or cancer cells. They recognize antigens presented on MHC class I molecules, which all nucleated cells in your body possess. When a cell is infected with a virus, it displays fragments of viral proteins on these MHC molecules, essentially showing T cells what's happening inside. If a cytotoxic T cell recognizes viral antigens, it confirms the cell is infected and delivers lethal enzymes that trigger the cell's programmed death. This eliminates the infected cell before viruses can replicate and spread, helping contain infections. The interaction between cytotoxic T cells and their targets is highly specific and carefully controlled. The T cell must recognize not only its specific antigen, but also verify that the target cell is truly infected through multiple checkpoints. This prevents cytotoxic T cells from accidentally killing healthy cells. When all conditions are met, the T cell forms a tight junction with the target cell and releases its lethal cargo directly into the target, minimizing collateral damage to surrounding cells. Regulatory T cells, which we mentioned earlier, provide crucial checks and balances on adaptive immunity. They suppress excessive immune responses, helping shut down reactions once a threat is eliminated. They also maintain tolerance to harmless antigens and to beneficial microbes, preventing inappropriate immune activation. The importance of regulatory T cells is demonstrated by what happens when they're absent or defective. Severe, often fatal autoimmune diseases that begin in infancy. One of adaptive immunity's most remarkable features is its ability to adapt. Not just during your lifetime, but during a single immune response. B cells undergo a process called affinity maturation in specialized structures within lymph nodes, called germinal centers. As B cells divide and produce antibodies, they also introduce random mutations into their antibody genes. Most mutations are harmful, producing antibodies that don't work as well. But occasionally, a mutation produces an antibody that binds the antigen more tightly. B cells producing these improved antibodies receive stronger signals from their receptors, encouraging them to survive and divide while cells with worse antibodies die off. Through multiple rounds of this selection process, the average quality of antibodies improves dramatically over the course of weeks, becoming better and better at binding and neutralizing the pathogen. This is one reason why antibody responses become more effective with time and why subsequent exposures to the same pathogen generate higher quality antibodies more quickly. The immune system is literally evolving in real time, using the same mechanisms of mutation and selection that drive biological evolution, but compressed into a much shorter time frame. B cells can also undergo class switching, changing which type of antibody they produce while maintaining the same antigen specificity. Different antibody classes are better suited for different types of threats. Some are particularly good at neutralizing pathogens in blood. Others are secreted into mucus or across the placenta. The signals that B cells receive from helper T cells and from the local environment determine which class they switch to, ensuring that the right type of antibody is produced for each situation. Adaptive immunity's sophistication comes at a cost. The initial activation of naive lymphocytes takes time, usually several days, because the few cells specific for a particular antigen must be located, activated, and clonally expanded. During this lag period, you rely entirely on innate immunity to hold off the infection. If innate immunity is overwhelmed before adaptive immunity comes online, the infection can become dangerous or even fatal. This is why people with defective adaptive immunity are vulnerable to infections that healthy individuals clear easily. But once adaptive immunity is fully engaged, its effects are dramatic. Antibody concentrations rise sharply. Cytotoxic T. Cells proliferate and begin destroying infected cells. Helper T cells coordinate multiple aspects of the response. The pathogen, which might have been multiplying freely despite innate immune efforts, suddenly faces a multi-pronged, highly specific attack. Most often, this tips the balance decisively in your favor, allowing you to clear the infection and begin recovering. The interplay between innate and adaptive immunity is crucial. Innate immunity doesn't just hold the line while adaptive immunity activates. It actively shapes the adaptive response. The danger signals detected by innate immune cells determine whether adaptive immunity is activated at all. The types of pathogens detected and the cytokines released by innate immune cells influence which types of helper T cells develop, which in turn affects the overall character of the immune response. And adaptive immunity can enhance innate immunity, with antibodies making phagocytosis more efficient, and T cells activating macrophages to kill intracellular pathogens more effectively. This cooperation between systems, each compensating for the other's limitations, produces immune defense that is both rapid and specific, both flexible and powerful. Together, they provide a layered defense that can handle almost any threat, from the commonplace to the exotic. And through immunological memory, which we'll explore next, the system becomes ever more effective, building a library of past defenses that can be deployed almost instantly when old enemies return. The gift of memory, how your body remembers. Perhaps the most consequential feature of adaptive immunity is its ability to remember. Once your immune system has encountered and defeated a pathogen, it retains the ability to respond to that pathogen much more quickly and effectively if it ever appears again. This immunological memory is the reason you typically get diseases like chickenpox or measles only once, even though you remain exposed to these viruses throughout your life. It's also the principle that makes vaccination possible, allowing you to gain immunity to dangerous diseases without ever experiencing them. Immunological memory is created during the primary immune response, your body's first encounter with a new antigen. As we discussed, this initial response takes time to develop, usually several days. During clonal expansion, as antigen-specific B and T cells multiply, most of the newly formed cells differentiate into effector cells, specialized for immediate combat. These cells, plasma cells secreting antibodies or cytotoxic T cells destroying infected cells, fight the current infection with intense activity but relatively short lifespans. Once the pathogen is eliminated, most effector cells die off, no longer needed. But not all the cells produced during clonal expansion become effector cells. A subset differentiates instead into memory cells, long-lived lymphocytes that persist after the infection resolves. Memory B cells continue to circulate through blood and lymphatic tissues, or take up residence in specific organs, maintaining the ability to quickly become plasma cells if they encounter their antigen again. Memory T cells do likewise, patrolling the body or residing in tissues where they first encountered their antigen, ready to reactivate if needed. Memory cells differ from naive cells in important ways. They're more numerous than naive cells specific for the same antigen, thanks to clonal expansion. They're more easily activated, requiring less stimulation to spring into action. And they're often already partially activated, having undergone changes during their initial differentiation that prepare them for rapid response. Some memory T cells even take up residence in tissues like the skin, lungs, and intestines, positioning themselves at likely sites of reinfection where they can respond immediately. When memory cells encounter their antigen again, the result is a secondary immune response that's dramatically faster and stronger than the primary response. Instead of days, antibody production can begin within hours. Instead of modest antibody levels, concentrations can skyrocket to levels far exceeding those seen during the primary response. And the antibodies themselves are often higher quality, thanks to affinity maturation during the first infection. Memory cytotoxic T cells can also respond rapidly, quickly destroying infected cells and helping contain infection before it becomes symptomatic. This accelerated response is the reason why many infections make you sick only once. When you're first exposed to a virus like measles, your immune system takes over a week to fully activate. During that time, the virus multiplies and spreads, causing the characteristic rash and symptoms. But your immune system eventually defeats the infection, and memory cells persist. If you're ever exposed again, memory cells respond so quickly that the virus is eliminated before it can multiply enough to cause illness. You might not even realize you were exposed. The duration of immunological memory varies depending on the pathogen and the individual. For some infections, memory persists for life. People who survived smallpox as a children retained immunity into old age, even though the disease was eradicated decades ago, and they had no subsequent exposures. For other infections, memory gradually wanes over years or decades. This is why you can get flu multiple times, though in that case, viral evolution also plays a role, with new flu strains sufficiently different from old ones that your existing memory doesn't fully protect you. Scientists are still working to understand what determines the longevity of immunological memory. Some memory cells are intrinsically long-lived, surviving for years or decades through mechanisms that aren't fully understood. Other memory cells may require occasional re-stimulation to persist. There's evidence that some memory cells are maintained by periodic re-exposure to antigens, perhaps from subclinical infections, or from residual antigen trapped in lymphoid organs. The question of whether memory cells are autonomous or require antigen to persist has important implications for vaccine design and for understanding waning immunity. Some memory cells, particularly certain plasma cells, take up residence in the bone marrow where they continue secreting antibodies indefinitely, providing constant protection without requiring re-stimulation. These long-lived plasma cells can maintain detectable antibody levels for years or even decades after a single infection or vaccination. This persistent antibody presence provides immediate protection, neutralizing pathogens before they can establish infection, while memory B and T cells stand ready to mobilize if antibody levels are insufficient. Memory formation is the basis of vaccination, one of medicine's greatest achievements. Vaccines work by exposing your immune system to antigens from dangerous pathogens in a safe context without causing disease. This allows your immune system to mount a primary response, generate memory cells, and prepare for future encounters, all without the risks of actual infection. The antigens in vaccines might be killed or weakened pathogens, purified components of pathogens, or even just genetic instructions for your cells to temporarily produce pathogen proteins. When you receive a vaccine, your immune system responds much as it would to actual infection, activating B and T cells specific for the vaccine antigens. Because the vaccine doesn't cause disease, you might experience only minor symptoms, if any, while your immune system builds memory. Then, if you're ever exposed to the real pathogen, your memory cells respond rapidly, often eliminating the infection before it causes symptoms. You gain immunity without suffering through the disease. Some vaccines provide protection after a single dose, while others require multiple doses spaced weeks or months apart. This isn't arbitrary. Booster doses take advantage of immunological memory, exposing the immune system to the antigen again after memory cells have formed. This triggers a secondary response, further expanding the memory cell population and improving antibody quality through additional rounds of affinity maturation. The result is stronger, longer-lasting immunity than a single dose would provide. For certain diseases, particularly those caused by rapidly evolving pathogens like influenza, periodic booster vaccinations are necessary to maintain immunity. This might be because the pathogen has changed enough that existing memory doesn't fully recognize it, or because immune memory to that pathogen naturally wanes over time. Understanding why some vaccines provide lifelong immunity, while others require boosters, is an active area of research with important practical implications. Interestingly, recent research has revealed that immunological memory might be distributed throughout your body in ways previously unappreciated. While memory cells in blood have been studied extensively, scientists now know that many memory cells reside in tissues, particularly in organs where pathogens commonly enter like the lungs and intestines. These tissue-resident memory cells provide localized protection, responding immediately to infections at those sites without requiring cells to migrate from distant lymphoid organs. This discovery has changed how scientists think about immune memory and may influence future vaccine designs. There's even evidence that the innate immune system can form a kind of memory, though it's quite different from adaptive immunological memory. After encountering certain pathogens, innate immune cells can undergo epigenetic changes that make them respond more vigorously to subsequent infections, even infections with different pathogens. This trained immunity is less specific than adaptive memory and doesn't last as long, but it provides a form of enhanced defense that bridges innate and adaptive immunity. The study of immunological memory continues to reveal new complexities. Scientists have discovered that memory cells are not uniform, but include multiple subsets with different properties and locations. Some memory cells are poised for rapid antibody production or cytokine secretion. Others are longer-lived but slower to respond. The relative proportions of these subsets and how they're maintained over time affects the quality and duration of immunity. One of the puzzles of immunological memory is its remarkable longevity in the face of constant cell turnover. Most cells in your body have finite lifespans and are regularly replaced. Yet some memory cells can persist for decades, much longer than the typical lifespan of lymphocytes. This could occur through intrinsic mechanisms that extend cell survival, through slow self-renewal, where memory cells occasionally divide to maintain their numbers, or through periodic re-stimulation by persisting antigens. The answer likely varies depending on the cell type and the pathogen in question. As you lie there tonight, memory cells from every infection you've ever had, every vaccine you've ever received, are distributed throughout your body. In your bone marrow, long-lived plasma cells are quietly secreting antibodies against pathogens you might not have encountered in years. In your lymph nodes and spleen, memory B and T cells are circulating, ready to respond if their specific antigens appear. In your lungs, skin, and gut, tissue-resident memory cells stand guard at potential entry points. This distributed network of memory represents your immunological autobiography, a living record of your encounters with the microbial world, protecting you from dangers you've already faced and preparing you for challenges to come. Chemical messengers, cytokines, and communication. The immune system, for all its cellular complexity, would be ineffective without precise coordination. Immune cells must communicate constantly, alerting each other to danger, recruiting help, coordinating attacks, and knowing when to stand down. This communication occurs primarily through cytokines, a diverse family of small proteins that serve as the immune system's chemical messengers. Cytokines are produced by many types of cells, both immune and non-immune, and they affect cellular behavior in myriad ways. Each cytokine binds to specific receptors on target cells, triggering signaling cascades that alter gene expression and cellular activity. Some cytokines act locally, affecting only nearby cells. Others enter the bloodstream and have systemic effects throughout the body. The same cytokine can have different effects depending on which cell type it acts on, the dose, and the presence of other cytokines, creating a communication network of remarkable subtlety. Scientists have identified hundreds of different cytokines, organized into families based on their structures and functions. Interleukins, literally between white cells, facilitate communication among leukocytes. Dozens of interleukins have been discovered, each with specific roles. Some promote inflammation, recruiting immune cells to infection sites and activating them. Others suppress inflammation, helping resolve immune responses once threats are eliminated. The balance between pro-inflammatory and anti-inflammatory interleukins is crucial for healthy immune function. Interferons are cytokines named for their ability to interfere with viral replication. When cells become infected with viruses, they produce interferons that warn neighboring cells of danger. These neighboring cells respond by entering an antiviral state, expressing proteins that inhibit viral reproduction. This limits viral spread, giving adaptive immunity time to activate. Interferons also enhance the activity of NK cells and cytotoxic T cells, strengthening cellular defenses against infected cells. Chemokines are specialized cytokines that create chemical gradients guiding cell movement. When infection or injury occurs, cells at the site release chemokines that diffuse outward, forming a concentration gradient. Immune cells like neutrophils and monocytes can detect these gradients through chemokine receptors on their surface. They then follow the gradient toward higher concentrations, a process called chemotaxis, leading them directly to where they're needed. Different chemokines attract different cell types, allowing precise recruitment of specific defenders. Tumor necrosis factor, or F, is a particularly potent pro-inflammatory cytokine involved in the acute phase response to infection. Its name derives from early observations that it could cause tumor shrinkage, though its primary role is in coordinating inflammatory responses. TNF activates endothelial cells lining blood vessels, making them express adhesion molecules that help immune cells exit the bloodstream and enter tissues. It also activates macrophages and promotes fever and acute phase protein production by the liver. While TNF is crucial for fighting infection, excessive or chronic TNF production contributes to inflammatory diseases, making it a target for therapeutic intervention. Colony-stimulating factors regulate the production of white blood cells in bone marrow. During infection, these cytokines signal the bone marrow to ramp up production of neutrophils, monocytes, or other cell types, ensuring adequate supplies for the ongoing immune response. This is why your white blood cell count increases during infection. It reflects the bone marrow responding to cytokine signals by producing more immune cells. Transforming growth factor beta, or TGF-Y, is an immunosuppressive cytokine that helps maintain tolerance and prevent excessive inflammation. It's produced by regulatory T cells and other cell types, and it suppresses the activation and proliferation of many immune cells. TGF-B is crucial for preventing autoimmunity and limiting collateral damage from immune responses, but it can also be problematic in cancer, where it can suppress anti-tumor immunity. The effects of cytokines are not just additive but combinatorial. The presence of one cytokine can alter how cells respond to others, creating a complex network of interactions. Helper T cells provide a clear example. Different subsets of helper T cells are characterized by the cytokines they produce, and these cytokines polarize other immune cells toward particular response patterns. One subset promotes antibody responses and defense against extracellular pathogens. Another promotes macrophage activation and defense against intracellular bacteria. Yet another promotes inflammation useful against parasites. Which subset develops depends on the cytokines present during T cell activation, which in turn depends on what type of pathogen triggered the innate immune response. This means that the nature of the initial infection influences the entire character of the immune response, ensuring that defenses are tailored to the threat. Bacterial infections typically trigger responses that activate phagocytes and promote antibody production. Viral infections trigger strong interferon responses and cytotoxic T cell activation. Parasitic infections trigger allergic-type responses with mast cell activation and eosinophil recruitment. The cytokine network coordinates these different response patterns. Cytokines also mediate the systemic effects of infection, the symptoms that make you feel sick. When you have the flu and experience fever, fatigue, muscle aches, and loss of appetite, you're feeling the effects of cytokines. Fever results from interleukin-1 and TNF, acting on the hypothalamus. Fatigue and malaise reflect cytokines acting on the nervous system. These symptoms, while unpleasant, are not merely side effects but adaptive responses. Fever inhibits pathogen growth. Fatigue encourages rest, directing energy toward immune defense rather than activity. Loss of appetite prevents iron and zinc from being absorbed, and these nutrients fuel bacterial growth. Your symptoms reflect your immune system actively combating infection. However, excessive cytokine production can be harmful. In severe infections, a phenomenon called cytokine storm can occur, where positive feedback loops cause uncontrolled escalation of cytokine production and inflammation. The resulting tissue damage can be worse than the infection itself, potentially causing organ failure and death. This is one reason why some severe infections are so dangerous. They trigger immune responses so vigorous that they harm the host. Regulating cytokine production to achieve effective defense without excessive inflammation is a constant balancing act. Cytokine dysregulation also underlies many chronic diseases. Autoimmune diseases often involve inappropriate or excessive production of pro-inflammatory cytokines. Allergies reflect exaggerated responses to harmless antigens, mediated partly by cytokines. Some cancers produce immunosuppressive cytokines that inhibit anti-tumor immunity. Modern medicine has developed therapies targeting specific cytokines, either blocking them to reduce excessive inflammation or supplementing them to boost immune responses, demonstrating the central importance of these molecules in health and disease. The cytokine network also exhibits remarkable redundancy and pleiotropy. Redundancy means that multiple cytokines can often accomplish similar tasks, providing backup systems that prevent single points of failure. Pleiotropy means that single cytokines can have multiple, sometimes opposite, effects depending on context. This makes the system robust but also complex and sometimes difficult to predict. Recent research has revealed that cytokine signaling is not simply a matter of one cell sending signals to another. Cells can also respond to their own cytokines in autocrine loops, or to cytokines from nearby cells of the same type in paracrine loops. The timing and duration of cytokine exposure matters as much as which cytokines are present. Transient exposure can cause different effects than sustained exposure. The local concentration can determine whether a cytokine activates or inhibits a response. All of these nuances contribute to the precision with which the immune system operates. Understanding cytokines has been crucial for developing new medical treatments. By blocking specific cytokines or their receptors, doctors can treat autoimmune diseases and inflammatory conditions. By administering certain cytokines, they can boost immune responses in cancer treatment or improve outcomes in certain infections. The development of these therapies has also taught us much about immune regulation, revealing which cytokines are critical for which functions. As you rest tonight, your cells are engaged in constant conversation through cytokines. In the absence of infection, low-level cytokine signaling maintains immune homeostasis, keeping cells primed but not overly activated. In areas experiencing minor inflammation from small cuts or irritations you're not even aware of. Local cytokine gradients are coordinating defensive responses. And throughout your body, regulatory cytokines are maintaining the balance between vigilance and restraint, ensuring your immune system remains ready for threats without attacking your own tissue. This chemical dialogue, invisible and unconscious, is as essential to your survival as the immune cells themselves. When things go wrong, immune system disorders. The immune system's power and complexity create opportunities for malfunction. When this defense network fails or misbehaves, the consequences can range from inconvenient to fatal. Understanding immune system disorders provides insight not only into disease, but also into how the healthy system normally works. By examining what happens when things go wrong, we learn to appreciate what must go right every day. Immunodeficiency disorders occur when part of the immune system is absent or not functioning properly. These conditions leave people vulnerable to infections that healthy immune systems handle easily. Immunodeficiencies can be congenital, present from birth due to genetic defects, or acquired later in life due to disease, malnutrition, or medical treatments. Primary immunodeficiencies are genetic disorders affecting immune system development or function. More than 100 different primary immunodeficiencies have been identified, affecting various components of immunity. Some impair antibody production, leaving people susceptible to bacterial infections of the respiratory tract and sinuses. Others affect T-cell function, causing vulnerability to viral and fungal infections. Still others disrupt innate immunity, causing problems with phagocyte function or complement activation. The specific pattern of infections a person experiences often provides clues about which part of their immune system is defective. Severe combined immunodeficiency, or SCID, represents the most serious form of primary immunodeficiency. Infants with SCID have profound defects in both T and B cell function, leaving them vulnerable to all types of infections. Without treatment, usually bone marrow transplantation to restore immune function, these children rarely survive infancy. SCID gained public awareness through the story of David Vedder, the boy in the bubble, who lived in a sterile plastic enclosure to protect him from infections. Fortunately, early diagnosis through newborn screening now allows treatment before severe infections occur, dramatically improving outcomes. Acquired immunodeficiencies are more common than primary ones. HIV/AIDS is the most well-known acquired immunodeficiency. HIV specifically targets helper T cells, which are crucial for coordinating adaptive immune responses. As these cells are destroyed over time, the immune system becomes progressively weaker, eventually leaving people vulnerable to opportunistic infections and cancers that healthy immune systems easily control. Modern antiretroviral therapy can now suppress HIV replication, preserving immune function and allowing people with HIV to live near normal lifespans. But without treatment, AIDS is ultimately fatal. Other causes of acquired immunodeficiency include malnutrition, which impairs immune cell production and function, immunosuppressive medications used to prevent organ transplant rejection or treat autoimmune diseases, and cancer treatments like chemotherapy, which can destroy rapidly, dividing immune cells along with cancer cells. Even aging causes a gradual decline in immune function, termed immunosenescence, which is why elderly people are more vulnerable to infections and respond less well to vaccinations. Autoimmune diseases represent the opposite problem: excessive or misdirected immune responses that attack the body's own tissues. Despite mechanisms designed to prevent this, autoimmunity is surprisingly common, affecting roughly 5 to 8% of the population. Dozens of autoimmune diseases have been identified, each characterized by immune attack on specific tissues or organs. Type 1 diabetes results from autoimmune destruction of insulin-producing beta cells in the pancreas. Without insulin, cells can't take up glucose from the blood, leading to dangerously high blood sugar levels. People with type 1 diabetes require lifelong insulin replacement. The disease typically begins in childhood or adolescence, though it can occur at any age. Rheumatoid arthritis involves immune attack on the joints, particularly the synovial membranes lining joint spaces. Chronic inflammation damages cartilage and bone, causing pain, swelling, and eventually joint deformity and disability. The disease also causes systemic effects, including fatigue and increased cardiovascular risk. Multiple sclerosis results from immune cells attacking the myelin sheaths that insulate nerve fibers in the brain and spinal cord. This disrupts nerve signal transmission, causing symptoms ranging from numbness and vision problems to paralysis and cognitive impairment. The disease typically follows a relapsing-remitting course, with periods of worsening symptoms alternating with partial recovery. Systemic lupus erythematosus, or lupus, involves production of autoantibodies that attack multiple tissues throughout the body. Symptoms vary widely depending on which organs are affected, but can include joint pain, skin rashes, kidney disease, and inflammation of the heart and lungs. The disease predominantly affects women and varies greatly in severity. What causes autoimmunity remains incompletely understood. Genetic factors clearly play a role. Many autoimmune diseases run in families. Certain genetic variations affecting immune regulation are more common in people with autoimmunity. Environmental triggers also matter. Infections, medications, and other exposures can sometimes precipitate autoimmune disease in genetically susceptible individuals. Some researchers propose that molecular mimicry plays a role, where antibodies generated against a pathogen cross-react with similar molecules on human cells, initiating autoimmune attack. Disrupted regulatory T-cell function might also contribute by failing to suppress self-reactive lymphocytes that normally escape elimination. Treatment of autoimmune diseases typically involves immunosuppression, using medications to dampen excessive immune responses. This can be challenging because suppressing immunity broadly increases infection risk. Newer, more targeted therapies attempt to selectively inhibit the specific immune pathways driving disease while preserving other immune functions. These biologics, engineered proteins that block particular cytokines or cell types, have revolutionized treatment for many autoimmune diseases, though they're expensive and not effective for everyone. Allergies represent yet another type of immune dysfunction. Inappropriately vigorous responses to harmless substances. Allergens like pollen, pet dander, or certain foods are mistakenly recognized as threats, triggering immune responses that cause more harm than the allergen itself could ever cause. Most allergies involve a type of antibody called IgE, which normally defends against parasitic worms. When IgE binds to an allergen, it activates mast cells and basophils, causing them to release histamine and other inflammatory mediators. This produces the familiar symptoms of allergic reactions: sneezing, runny nose, itchy eyes in hay fever, wheezing, and shortness of breath in asthma, hives and swelling in food allergies. In severe cases, massive mast cell activation can cause anaphylaxis, a life-threatening reaction involving airway constriction, circulatory collapse, and potentially death without immediate treatment with epinephrine. The prevalence of allergies has increased dramatically in recent decades, particularly in developed countries. This has led to the hygiene hypothesis, which proposes that reduced childhood exposure to microbes due to improved sanitation and antibiotic use prevents normal immune system maturation, predisposing people to allergies. While this hypothesis remains debated, it's clear that environmental factors influence allergy development. Food allergies have also become more common, affecting millions of people. Common culprits include peanuts, tree nuts, shellfish, eggs, milk, wheat, and soy. Most children outgrow milk, egg, and wheat allergies, but peanut, tree nut, and shellfish allergies typically persist lifelong. The only current treatment is strict avoidance, though experimental therapies attempting to induce tolerance are under development. Chronic inflammatory diseases often involve immune dysfunction. Inflammatory bowel diseases like Crohn's disease and ulcerative colitis result from inappropriate immune activation in the gut, causing chronic intestinal inflammation, abdominal pain, and diarrhea. These conditions likely involve complex interactions between genetic susceptibility, alterations in gut bacteria, and immune dysregulation. Even conditions not traditionally considered immune disorders involve immune components. Cardiovascular disease involves chronic inflammation of blood vessel walls. Type 2 diabetes is associated with inflammatory changes in fat tissue. Cancer development is influenced by immune surveillance and by cancer cells' ability to evade or suppress immunity. The immune system's influence extends far beyond fighting infection, affecting health and disease in surprising ways. The balance required for healthy immune function is delicate. The system must be powerful enough to fight infections, but restrained enough to avoid attacking the body itself or overreacting to harmless exposures. It must distinguish pathogens from beneficial microbes, cancerous cells from healthy ones, foreign proteins from self-proteins. It must generate vigorous responses when needed, but then resolve them when threats are eliminated. Achieving this balance requires multiple layers of regulation, redundant safety mechanisms, and constant calibration. When we appreciate how many things must go right for the immune system to function properly, it's perhaps surprising that immune disorders aren't more common. The system's robustness, built through hundreds of millions of years of evolution, and refined through each individual's lifetime of antigen exposures, usually maintains the balance successfully. But when it fails, whether through deficiency, excessive activity, or misdirection, The consequences remind us of how dependent we are on this hidden network of defense and how much we still have to learn about its workings. Lifestyle and immunity, the connection. Your immune system doesn't operate in isolation from the rest of your life. How you sleep, what you eat, your stress levels, your physical activity. All of these aspects of daily life influence immune function in profound ways. Understanding these connections empowers you to support your immune health through lifestyle choices. Sleep and immunity are intimately linked. During sleep, your immune system performs maintenance tasks and consolidates immunological memory. Sleep deprivation impairs immune function in multiple ways, reducing the numbers of certain immune cells, weakening responses to vaccination, and increasing susceptibility to infection. Studies have shown that people who regularly sleep less than 7 hours per night are more likely to catch colds when exposed to viruses compared to those who sleep 8 hours or more. The relationship works both ways. Immune activation influences sleep patterns. During infections, increased levels of certain cytokines promote sleep, particularly deep, slow-wave sleep. This is why you feel exhausted and sleep more when you're sick. The sleep helps your immune system fight the infection more effectively. Interfering with this sleep, staying active when your body is urging rest, can prolong illness. Sleep's immune benefits likely reflect multiple mechanisms. During deep sleep, levels of stress hormones like cortisol drop, while levels of growth hormone and prolactin increase. These hormonal shifts favor immune cell proliferation and the formation of immunological memory. Sleep also provides an opportunity for the body to redirect energy toward immune functions, rather than supporting waking activities. The immune system appears to have evolved to take advantage of sleep's quiescent state to perform resource-intensive tasks. Chronic sleep deprivation has been linked to increased inflammation detectable through elevated levels of inflammatory markers in the blood. This chronic low-grade inflammation is thought to contribute to various health problems, including cardiovascular disease, metabolic disorders, and even cognitive decline. The immune system, designed for acute responses to threats, can cause harm when chronically activated. Nutrition profoundly affects immunity. Protein deficiency impairs immune cell production and function because antibodies and many immune molecules are made of protein. Deficiencies of specific vitamins and minerals can also compromise immunity. Vitamin C, for instance, supports various immune functions and is used in high amounts by immune cells during infections. Vitamin D helps regulate immune responses and appears particularly important for defense against respiratory infections. Zinc is essential for immune cell development and signaling. Severe malnutrition causes obvious immune impairment, increasing vulnerability to infections. But even marginal deficiencies of specific nutrients, common in developed countries, can subtly affect immune function. This is why eating a varied, nutrient-rich diet supports immune health better than relying on supplements, though supplementation may be beneficial when dietary intake is insufficient. The gut microbiome, the trillions of bacteria living in your intestines, also profoundly influences immunity. These microbes help train the immune system during development. interact constantly with immune cells in the gut and produce metabolites that affect immune function throughout the body. Disruption of the microbiome through antibiotics, poor diet, or other factors can have immunological consequences. Fiber, found in fruits, vegetables, and whole grains, supports beneficial gut bacteria, which ferment it into short-chain fatty acids. These compounds have anti-inflammatory effects and help maintain the integrity of the intestinal barrier, preventing unwanted substances from leaking into the bloodstream where they could trigger inappropriate immune responses. Probiotic foods like yogurt and fermented vegetables may also benefit immune function by promoting helpful bacterial populations, though the science here is still developing. Obesity affects immunity in complex ways. Fat tissue isn't just for storage. It's metabolically active, producing hormones and inflammatory cytokines. Excess fat, particularly abdominal fat, creates a state of chronic low-grade inflammation that can impair some immune functions while exacerbating others. People with obesity often experience higher rates of certain infections and less effective responses to vaccinations. They're also more prone to inflammatory conditions. Weight loss can improve immune function, though the relationship is complicated by nutritional status and other factors. Stress, both acute and chronic, significantly impacts immunity. Short-term stress can actually enhance certain immune responses, a vestige of the fight-or-flight response where the body prepares for potential injury and infection. However, chronic stress suppresses immune function, increasing vulnerability to infections and slowing wound healing. This occurs partly through elevated cortisol, a stress hormone that has anti-inflammatory effects. While helpful in the short term, sustained cortisol elevation impairs immune cell proliferation and function. Chronic stress also affects health behaviors. Stressed people often sleep poorly, eat less nutritious foods, exercise less, and may use alcohol or other substances to cope, all of which further compromise immunity. The psychological and physiological aspects of stress thus combine to weaken immune defense. Importantly, stress effects aren't just psychological. Social isolation, loneliness, and lack of social support are associated with altered immune function and increased inflammation. Social connections appear to benefit health partly through immune mechanisms, though the pathways involved remain incompletely understood. This underscores that immunity is influenced not just by biology, but by social and psychological factors. Exercise benefits immunity in multiple ways. Moderate, regular exercise enhances immune surveillance, reduces inflammation, and improves responses to vaccination. Each bout of exercise causes immune cells to circulate more rapidly through the body, improving their ability to detect threats. Over time, regular exercise appears to slow age-related decline in immune function. However, very intense or prolonged exercise can temporarily suppress immunity, creating an open window of increased infection vulnerability lasting several hours after exertion. Elite athletes who train very hard often experience more respiratory infections than moderately active people. The key is finding the right balance. Regular, moderate exercise supports immunity, but excessive training without adequate recovery can be counterproductive. Age profoundly affects immune function. The immune system changes throughout life, from the highly active, developing system of childhood through the robust immunity of adulthood to the gradual decline of aging. Newborns have immature immune systems, depending partly on antibodies transferred from their mothers. Immune function matures through childhood as they're exposed to pathogens and develop memory. By adulthood, most people have robust immunity shaped by years of exposures. With aging, immune function gradually declines, a process called immunosenescence. The thymus shrinks, producing fewer new T cells. The population of naive lymphocytes capable of recognizing new pathogens decreases while memory cells, specific for pathogens encountered earlier in life, accumulate. Innate immunity changes too, with altered patterns of inflammatory signaling. These changes mean elderly people respond less effectively to new infections and to vaccinations, and they may experience more severe complications from infections. Chronic low-grade inflammation, sometimes called inflammaging, also increases with age, contributing to age-related diseases. However, immunosenescence isn't uniform or inevitable. Lifestyle factors including nutrition, exercise, sleep, and stress management can slow immune aging. People who maintain healthy lifestyles often retain better immune function into old age than those who don't. Smoking damages immunity in multiple ways. It impairs the function of cilia in the respiratory tract, reduces the activity of immune cells in the lungs, and causes chronic inflammation. Smokers are more susceptible to respiratory infections and take longer to recover. Smoking also affects systemic immunity, not just the lungs. Quitting smoking allows some recovery of immune function, though some damage may be permanent. Alcohol also affects immunity. Moderate consumption may have neutral or even slightly beneficial effects in some contexts, but heavy drinking clearly impairs immune function. Alcohol disrupts gut barrier function, alters the microbiome, impairs the function of immune cells, and increases inflammation. People with alcohol use disorder have higher rates of infections, particularly pneumonia and tuberculosis. Understanding these lifestyle connections offers opportunities to support immune health through daily choices. While you can't control your genetic predisposition to immune disorders or your exposure to all pathogens, you can influence how well your immune system functions through how you live. Adequate sleep, nutritious diet, regular moderate exercise, stress management, maintaining healthy weight, avoiding smoking, and limiting alcohol all contribute to resilient immunity. It's worth noting that no lifestyle intervention can completely prevent disease, and sometimes people do everything right and still get sick. Luck plays a role, as does genetics, but statistically, people who maintain healthy lifestyles tend to have fewer infections, less severe illness when they do get sick, and better responses to vaccinations. They also typically experience less chronic inflammation, which benefits long-term health beyond just immunity. As you settle into sleep tonight, your lifestyle choices over the past days and weeks have already influenced your immune function. If you've slept well, you're giving your immune system time to consolidate memory and perform maintenance. If you've eaten nutritious foods, you're providing the building blocks and fuel your immune cells need. If you've managed stress, exercised regularly, and avoided harmful substances, you've created conditions where your immune system can function optimally. These choices, accumulating over time, shape the invisible defensive network that protects you every day, the future. Emerging understanding. Immunology is a dynamic field, constantly revealing new aspects of how our bodies defend against disease. Recent years have brought discoveries that challenge traditional models and open new therapeutic possibilities. As we explore these emerging insights, we see how much remains to be learned about this vital system. One transformative discovery is tissue-resident memory cells. Scientists long assumed that memory lymphocytes circulated continuously through blood and lymph. But recent research has shown that many memory cells take up permanent residence in specific tissues. These tissue-resident memory T cells, or TRMs, remain in organs like the skin, lungs, and intestines where infections commonly occur, providing rapid, localized protection without requiring cells to travel from distant lymphoid organs. RMs have distinct properties compared to circulating memory cells. They were positioned exactly where they're most needed, can respond within hours of antigen reappearance, and are maintained independently of circulating lymphocyte populations. This discovery has important implications for vaccination. Traditional vaccines delivered into muscle primarily generate circulating immunity. Newer approaches aim to generate TRMs at likely infection sites, potentially providing superior protection against respiratory and mucosal pathogens. The concept of trained immunity, which we've mentioned before, represents another paradigm shift. The traditional view held that innate immunity, unlike adaptive immunity, lacks memory. But research has shown that innate immune cells can undergo epigenetic reprogramming after exposure to certain stimuli, making them respond more vigorously to subsequent challenges. This training doesn't involve antigen-specific receptors like adaptive memory, but rather broad changes in gene expression that enhance general responsiveness. Trained immunity has practical implications. The BCG vaccine, originally developed against tuberculosis, has been shown to reduce mortality from unrelated infections through trained immunity effects. Some vaccines may protect partly through training innate immunity rather than solely through adaptive memory. Understanding these mechanisms could enable development of new vaccines and immunomodulatory treatments that harness trained immunity. The microbiome's influence on immunity is another area of intense investigation. The trillions of microbes colonizing our bodies, particularly in the gut, profoundly affect immune development and function. Germ-free animals raised without any microbes have severely impaired immunity. In humans, differences in microbiome composition correlate with susceptibility to infections, autoimmune diseases, and allergies. The relationship is bidirectional. The immune system shapes the microbiome by selectively tolerating beneficial bacteria while eliminating pathogens. The microbiome, in turn, influences immune development, helps train the immune system to distinguish friends from foes, and produces compounds that affect immunity throughout the body. Disrupting this relationship through antibiotics, diet changes, or other factors can have immunological consequences. This understanding is driving new therapeutic approaches. Fecal microbiota transplantation, transferring gut bacteria from healthy donors to patients, has shown dramatic success in treating certain infections and is being explored for other conditions. Dietary interventions and prebiotics that favor beneficial bacteria are being studied as ways to modulate immunity through the microbiome. The field of psychobiotics even investigates how gut bacteria influence brain function and stress responses, which in turn affect immunity, creating a fascinating gut-brain immune axis. Cancer immunotherapy represents one of the most exciting applications of immunological understanding. For decades, scientists have known that the immune system can recognize and attack cancer cells, but cancers evolve mechanisms to evade or suppress immunity. Modern immunotherapies aim to overcome these evasion strategies, unleashing the immune system against tumors. Checkpoint inhibitor drugs block proteins that cancers use to switch off T-cell attacks. By removing these breaks, the drugs allow T-cells to attack tumor cells more effectively. This approach has produced remarkable, durable responses in some patients with previously untreatable cancers, though it also causes autoimmune-like side effects in many patients by disrupting normal immune regulation. CAR T-cell therapy genetically engineers a patient's own T-cells to recognize and attack their specific cancer. T-cells are collected, modified to express engineered receptors that target cancer cells, expanded in the laboratory, and then infused back into the patient. This living drug has cured some patients with leukemias and lymphomas that didn't respond to other treatments. Researchers are working to extend this approach to solid tumors and to make the therapy safer and more widely accessible. Cancer vaccines are also being developed, aiming to train the immune system to recognize tumor-specific antigens. Unlike traditional vaccines that prevent infections, cancer vaccines are typically therapeutic, treating existing cancers rather than preventing them. Personalized cancer vaccines, designed to target the specific mutations in a patient's tumor, are showing promise in clinical trials. Advances in vaccine technology have also emerged. The COVID-19 pandemic accelerated development of mRNA vaccines, which provide genetic instructions for cells to produce viral proteins, triggering immunity. This platform is faster to develop and manufacture than traditional vaccines and can be readily adapted to new pathogens. Similar approaches are being explored for other infectious diseases and even cancer. Nanoparticle vaccines deliver antigens in particles designed to be taken up efficiently by immune cells. These can include adjuvants, compounds that enhance immune responses, and can be engineered to target specific immune cells or lymphoid organs. Such precision could improve vaccine effectiveness while reducing side effects. Oral and inhaled vaccines are being developed to generate immunity at mucosal surfaces, potentially providing better protection against respiratory and gastrointestinal pathogens than injected vaccines. These routes also avoid needles, which could improve acceptance and simplify administration. Understanding of autoimmune disease mechanisms has advanced considerably. Rather than treating autoimmunity solely with broad immunosuppression, newer therapies target specific pathways driving particular diseases. Biologics, blocking specific cytokines, have transformed treatment of rheumatoid arthritis, inflammatory bowel disease, and psoriasis. Cell-based therapies that restore proper immune regulation are under investigation. Gene therapy might someday correct the underlying genetic defects that predispose to autoimmunity. Researchers are also working to induce antigen-specific tolerance, teaching the immune system to stop attacking specific self-antigens without broadly suppressing immunity. If successful, this could cure autoimmune diseases rather than merely controlling symptoms. Early trials using this approach in type 1 diabetes and multiple sclerosis have shown promise, though much work remains. The role of the immune system in aging and age-related diseases is increasingly recognized. Chronic inflammation associated with aging, inflammaging, contributes to cardiovascular disease, neurodegeneration, cancer, and frailty. Understanding and counteracting immunosenescence could help people remain healthier longer. Senolytics, drugs that eliminate senescent cells that accumulate with aging and contribute to inflammation, are being tested as anti-aging interventions with potential immunological benefits. Single-cell technologies are revolutionizing immunology research. Traditional methods studied bulk populations of cells, averaging out the differences between individuals. New technologies can analyze thousands or millions of individual cells, revealing previously hidden diversity and rare cell types. This granular view is exposing complexity in the immune system that scientists are only beginning to understand, including new cell subtypes, unusual activation states, and heterogeneous responses to pathogens. Systems immunology takes a holistic approach, using computational methods to analyze the complex interactions among immune components. Rather than studying individual cells or molecules in isolation, systems immunology examines networks, feedback loops, and emergent properties. This approach helps predict how immune responses will unfold and how interventions will affect the system, potentially enabling more rational vaccine and therapy design. Precision medicine in immunology aims to tailor treatments to individual patients based on their unique immune profiles, genetics, and disease characteristics. As we understand immune diversity better, we can move beyond one-size-fits-all approaches toward personalized immunotherapies that account for each patient's specific situation. Despite these advances, major questions remain. We still don't fully understand what determines the longevity of immunological memory, or how to reliably generate long-lasting immunity through vaccination. The mechanisms of autoimmune disease initiation remain incompletely explained. We're only beginning to grasp how the immune system communicates with other body systems and how factors like stress, diet, and sleep exert their effects. The immune system's role in chronic diseases beyond traditional immune disorders is still being explored. As immunology continues advancing, it's likely to transform medicine. Better vaccines could prevent or even cure diseases currently treated with drugs. Immunotherapies might offer new treatments for cancer, autoimmunity, and infections resistant to antibiotics. Understanding trained immunity and tissue-resident memory could enable new protective strategies. Harnessing the microbiome might provide natural ways to modulate immunity. The immune system, hidden and automatic as it is, touches every aspect of health. The more we learn about it, the more we realize how central it is to human biology, how elegant its solutions to the challenge of survival, and how much potential exists for therapeutic innovation. The coming decades will likely bring discoveries that change how we think about immunity and how we maintain health throughout life. Conclusion and closing. We've journeyed through the vast, hidden universe of your immune system. From the organs where immune cells are born and trained to the diverse cellular forces that patrol your body. We've witnessed the swift, ancient responses of innate immunity and the precise, sophisticated attacks of adaptive immunity. We've explored the gift of immunological memory that protects you from old enemies and makes vaccination possible. We've seen how chemical messengers coordinate this complex system and what happens when the delicate balance is disrupted. We've learned how your daily choices influence immune function, and we've glimpsed the frontiers of immunological research where new discoveries promise to transform medicine. But understanding the immune system is more than an intellectual exercise. It's a way of understanding yourself at the most fundamental level. Every scar you carry represents immune cells that rushed to wounds and orchestrated healing. Every infection you've survived left behind memory cells that still patrol your body. The iron in your blood was delivered by immune cells clearing old red blood cells. The proteins in your blood include antibodies, molecular sentinels produced by your immune system. You are not separate from this defensive network. You are it. Right now, as you lie there in the quiet, this system continues its endless work. In your bone marrow, stem cells are dividing, giving rise to new immune cells that will enter circulation tomorrow. In your lymph nodes, memory cells are quietly waiting, some remembering infections from decades ago. In your lungs and gut, tissue-resident cells stand watch at the frontiers where the inside meets outside. In your bloodstream, neutrophils are pursuing their brief, intense lives, ready to sacrifice themselves should infection threaten. And throughout your body, regulatory mechanisms are maintaining balance, ensuring vigilance without overreaction, defense without self-destruction. This system evolved over hundreds of millions of years, refined by countless generations facing the challenge of surviving in a world filled with microscopic threats. The result is a masterpiece of biological engineering, a distributed intelligence that operates without consciousness, learns without a brain, and remembers without a mind. It represents one of life's fundamental solutions to a fundamental problem: how to maintain integrity, how to remain yourself in a world constantly trying to invade, exploit, or alter you. Yet for all its sophistication, the immune system remains imperfect. It can fail, malfunction, or turn against you. It sometimes misses threats or attacks harmlessly stimuli. It weakens with age and is influenced by stress, sleep, and nutrition in ways we're only beginning to understand. But even in its imperfections, the system reflects the constraints under which it evolved, the trade-offs between power and restraint, the balance between responding to threats and avoiding harm. As science continues unraveling the immune system's mysteries, we gain not just knowledge but power, the power to prevent diseases through better vaccines, to treat cancers and autoimmune disorders through immunotherapy, to support immune health through informed lifestyle choices. We're learning to work with this system, to enhance its strengths and compensate for its weaknesses, to harness its power for healing. But perhaps most importantly, understanding immunity fosters appreciation. Appreciation for the countless battles fought on your behalf without your awareness. Appreciation for the elegant solutions evolution discovered to seemingly impossible problems. Appreciation for the resilience built into this system through redundancy, adaptability, and memory. And appreciation for the daily miracle of waking up healthy, a state made possible by trillions of cells working in concert to protect you. Tonight, as sleep claims you, your immune system will continue its watch. It will clear the cellular debris of the day. It will patrol for abnormalities. It will consolidate memories of any challenges encountered. It will maintain the peace with beneficial microbes while standing ready to repel invaders. And it will do all of this automatically, reliably, without any conscious direction from you. You are protected. Not perfectly, not without vulnerabilities, but protected nonetheless by one of nature's most remarkable inventions. An invisible army, a distributed intelligence, a guardian that never sleeps. Your immune system has been there since before you were born and will remain until the end, changing as you change, learning as you learn, protecting you from dangers you'll never know you faced. If this journey into immunology has given you new appreciation for your body's hidden defenses, or if these gentle explorations have helped you unwind and find peace at the end of your day, I'd be grateful if you'd take a moment to like this video and subscribe to the Sleepy Science Society. It helps others find these calm, curious adventures into science, one peaceful night at a time. Sleep well, knowing that as you rest, countless microscopic guardians stand watch. Dream softly, protected by systems so ancient and yet so sophisticated that science is still discovering their secrets. And rest in the knowledge that tomorrow, when you wake, your immune system will still be there, quietly doing what it has done every day of your life, keeping you alive, keeping you healthy, keeping you you. Good night.